Okay, this presentation is from a seminar that I taught about perfusion, and it's heavily focusing on the technical part and a little bit less about the clinical part because it's easier to read the clinical applications from papers and study them independently, whereas it's a bit harder to understand the technical part without a formal explanation. And then we also see some technical advancements. So just a disclaimer, we will talk about perfusion MRI in brain tumors and only the techniques that use contrast agent. So some concepts about brain tumors, DCE and DSC technical bases and leakage effects, some clinical applications, but that'd be very brief. Multi-echo perfusion in terms of what's new, what are possible advancements and why it could be useful, and the concept of pseudo-resting state. So first of all, some brain tumors show contrast enhancement. It means that from pre-contrast T1 weighted images to post-contrast T1 weighted images, there is a MR signal increase. This is due to the fact that there is some leakage in the due to disruption in the blood-brain barrier, so that there is leakage of contrast agent from the vascular compartment to the extravascular compartment. However, other tumors do not really show this leakage because the blood-brain barrier is still kind of intact, so they are not enhancing. Just bear in mind there are two different situations. I want to introduce what the concept of 4D imaging is. If we think about a conventional MRI of the brain, we can think about how the data is stored just like a 3D array. It means that in space, every little voxel has some coordinates in the XYZ space. So it's located somewhere in a three-dimensional space. So the intensity of the MR signal in a certain voxel is stored as part of a 3D array. This does not mean I'm talking about 3D imaging. 3D MRI refers, as a term, refers to how the MRI, what's the technical way of acquiring the images. But regardless of how you acquire 2D or 3D, whatever, you can still store MR images as 3D arrays. But then let's do a step further. There are some sequences that for which the data can be stored and has to be stored in 4 dimensions. So typically on top of a 3D dimension, there is a 4th dimension which can be time. So for instance, the brain that we talked about, if it's followed over time while I'm injecting contrast agent, there would be a 4th dimension. And for each voxel that has a certain XYZ coordinate in space, for each voxel, there's going to be a 4th dimension that tells you what the signal over time is and how it changes due to the contrast agent. Other advanced sequences can have 4th dimensions that are not strictly a temporal dimension. For instance, diffusion-weighted imaging can have different gradients, orientations of diffusion stored along a 4th dimension. But anyways, for what we're talking today, the 4th dimension is a time dimension. So let's talk about DCE, which is a T1 weighted technique. You see these are a serial T1 weighted image over time. So the tumor area, we see contrast enhancement that is progressive over time. So if we were to plot the intensity of the signal over time, we would see something like this, where there is a baseline, and then when contrast agent arrives within a certain voxel of the tumor, we can see an increase in the signal intensity. So this would be, for a given voxel in 3D space, the 4th dimension, where we see a intensity curve that is called time intensity curve, TIC. And overall, we can say that this curve, signal over time, reflects the contrast enhancement kinetics over time. This technique assumes leakage of the contrast from the vascular space to the extravascular space, and we call it a permeability imaging technique because it tells us something about the kinetics of the extravasation, of the leakage, of the contrast enhancement from the vascular to the extravascular space. So if we think about the concept that we said before, it's mainly for tumors that do show a contrast enhancement, because we are studying the kinetics of extravasation and leakage in those specific tumors, right? So if we look at the time intensity curve, we can already have some information by simply looking at this fourth dimension, because we know that there is a first early peak that has to do with the vascular component, whereas if the leakage is slower, there's going to be a more of a delayed signal intensity spike. And this is at the basis of what we call DCE curviology, where we classify curves of DCE as type 3 if there's an early spike, type 1 if there is a slow progressive enhancement, or type 2, which is in the middle. The biological correlates of these three types are not always super easy to understand, but this is a qualitative evaluation of the contrast enhancement kinetics over time if we look at the specific voxel or group of voxels in the fourth dimension. Using the time intensity curve, you can also derive some metrics that are called semi-quantitative metrics that can quantify certain aspects and characteristics of this curve. The most used of these metrics is IAUC 60, IAUC 30. What it means is that we calculate the integral, so the area under the curve of the time intensity curve from the bolus arrival, so from the contrast agent arrival to a certain amount of time, so 60 for 60 seconds, 30 for 30 seconds. And then since this is a single number, we can go back to a three-dimensional space, and we have for each voxel the single number that represents the area under the curve after, for instance, 60 seconds. I'm mentioning this because if you read DCE articles, you will see that they use sometimes AUC 60, AUC 30, so now you know what it is. But the further step, which is mostly used for DCE, is the pharmacokinetic modeling. What does that mean? So first of all, the signal over time curve that we saw can be converted into first a delta R1 over time curve, which is quantitative imaging. It means what's the longitudinal relaxation rate over time, actually the change in the longitudinal relaxation rate over time. I'm not showing it here, so it's a delta R1 over time curve. In order to convert signal to delta R1, you need to measure the T1 time of the tissue with T1 relaxometry, or you can assume a certain T1 of the tissue. So there's an assumption or a measurement involved. After you have delta R1, assuming another constant, which is little r1, we're not going to talk about that into detail, but we can convert delta R1 over time into a concentration curve. So at the end of the day, what I'm trying to say is that with some calculation, we can assume that the signal changes over time are proportional to the change in concentration over time. When I say concentration, I mean the concentration of the contrast agent, which is the blue curve here. So this is the change in concentration of the contrast agent that starts from 0 and then spikes over time. The pharmacokinetic modeling means that we are trying to fit a curve to get the parameters that we know and we usually use in DCE, like Ktrans and VE. What's important to understand is we have to do a curve fitting, which is the orange fit that you can see. So it's not the measured, as before, the measured signal or concentration over time. It's a fitted or modeled curve of the concentration over time. And the model converges given the measured concentration over time, given the concentration over time in an artery, which is here, CP, concentration, plasmatic concentration. It will converge to certain Ktrans and VE values. So the Ktrans and VE maps that we see are the result of the model fitting given the concentration over time in the voxel and the concentration over time in an artery. Why is that? Because these are the pharmacokinetic models that we use that assume a certain structure of the tissue. So what we just saw before, it's called the extended Tofts, which is the model that is usually used for brain tumors. The original Tofts didn't model the capillary space, like the plasmatic volume, let's say, because it considered it negligible. So he only modeled the Ktrans, the constant, sorry, not the constant, but the extravasation rate from the vascular to the extravascular component, and the KAP, which is the reflux rate from the extravascular space to the intravascular space, and then the amplitude of VE, which is the extravascular space. Now, the extended tufts that we talk about also has the vascular component. So it models VP, VE, Ktrans, KAP. This is a great model for brain tumors because it assumes a high degree of permeability, so a high constant of efflux from the intravascular to the extravascular space. And again, we need an arterial input function, which is a bit problematic. It's something that it's a bit of a headache to obtain. And most of all, the way you sample the intravascular curve does change, obviously, the way the model converges and does change your estimate of Ktrans. Finally, I want to mention the Patlack model. So just to understand how the results that we have, the Ktrans that we model, is dependent on the type of tissue structure that we are assuming. The Patlack model models differently because it assumes that the leakage of the blood-brain barrier is much, much slower. So it only models the Ktrans with no VE because if the extravasation is very slow over time, you can assume that the VE is pretty much infinite. And also that there is no reflux, there is no KAP because the concentration in DVE is always very small and negligible. So Patlack model is not it's not appropriate for brain tumors. It's usually used for those studies where you're studying subtle permeability, like let's see if the basal ganglia have subtle increased permeability in, let's say, Alzheimer's disease or something like that, where you cannot really see an actual evident apparent contrast enhancement, but you're studying subtle permeability. So for brain tumors, we use extended tufts, and we get Ktrans or V maps by fitting these models. These are some simulated curves from tufts, where you can see that like a higher Ktrans means more of a almost type 3 curve, so which with more early spike. It makes sense because it means that there's more extravasation in the unit of time. Let's remember Ktrans is minutes to the minus 1, so it's a rate. So more extravasation in the unit of time means that I'm going to see a more abrupt contrast enhancement in the voxel. Whereas if I have a larger VE, so a larger extravascular space, it's more of the tail of the curve that becomes higher and higher because there is more space for the contrast to extravasate without necessarily immediately flowing back into the intravascular space. Let's simplify and not talk about this in the specifics, but you see how also in MRI questions there are some schematics that show how Ktrans and VE changes affect the shape of the curve. Let's remember that KAP, Ktrans, and VE are intercorrelated. You can only change one at the time while keeping fixed the other one, but the third one is going to change accordingly with the first one that you're trying to change, of course. So it's not super easy. And all of these changes are assuming the same arterial input function, of course, and whatnot. One note about Ktrans, we usually say that Ktrans is a measure of permeability. This is correct to some extent. The Ktrans more correctly is the rate of extravasation from the intravascular to the extravascular space. Let's think about the subway on peak hours, like on rush hour, and let's say that the intravascular space is the train and the extravascular space is the station, right? If there is, so in presence of high flow, so a lot of people on rush hour, the Ktrans, which is the rate of passage from the train to outside the train, is limited by how many doors are open, which is it's limited from how much permeability the system has. So in presence of high flow, Ktrans reflects permeability. However, at 3 a.m. in the morning, there's no passengers, The doors can be open, so the system can be permeable, but since there's nobody flowing outside the train, the Ktrans will be low. So in that case, because of the blood flow, so the passengers in the train is very low as a number, it means that the Ktrans is limited by the flow and it doesn't reflect permeability anymore. The system can be permeable, but I have no way of measuring that. It's just as in a certain area of the brain, there is a lot of permeability of the blood-brain barrier, but there is very little blood getting there or very little contrast agent getting there. So in those cases, the K-trans doesn't necessarily reflect permeability. But in brain tumors, this is usually the scenario where there's a lot of blood and blood vessels. So the K-trans, we can say, reflects permeability. So overall, these are all the DCE maps, also some DSC maps. We'll see that later, including the quantitative maps like Ktrans, KPVE, and the semi-quantitative maps like area under the curve. What are the pros and cons of using quantitative or semi-quantitative? Quantitative are actual quantitative imaging. For instance, Ktrans is minutes to the -1, so it's a flow, and it accounts for personal variations of the arterial input function because that is in the formula. So it kind of adjusts for all of the variations in the injection of the contrast agent, the vascularity of the subject, and also some technical parameters like flip angle and all those things, right? So it should account for those things, but it has some assumptions. And so you have to pick the correct model to limit the impact of those assumptions and the error generated by those assumptions. But it's always approximated to some extent. And it's dependent on the arterial input function. As I said, it's a bit of a headache to get a correct arterial input function. And the way you sample it is going to change your K-trans results to some extent. The semi-quantitative measurements don't have these issues, but they don't correct for all of these variables. So since it's simply the area under the curve of the signal intensity, if you're changing the pulse sequence parameters such as the flip angle and whatnot, you're gonna change these measurements. And if you change the rate of injection, if the subject has a different vascular structure, it doesn't account for any of that. So pros and cons. Now let's talk about DSC.